o'clock, and I know people are still joining, and I want to welcome everyone to this morning's very special Grand Rounds presentation. For those of you in the Department of Pediatrics, this is a half hour earlier than we usually do rounds, so we can pretend we're in Newfoundland. But part of the reason we're doing this early is to accommodate our surgical colleagues and other colleagues in this special joint pediatrics surgical Grand Rounds. I will be your host for today in Dr. Jabour's absence. Before we start, I'm just going to share my screen to share some information about PSI, so hopefully this will work. Are you able to see that? Yes? Okay. So this morning is the leadoff speaker in our new PSI sponsored Visiting Scholar series focused on precision child health. We're very grateful to PSI for supporting this series and sponsoring Dr. Malkin's visit, and a big thanks to Jim King, who is our PSI representative, Heather Howley, who coordinates this series, and Dora Jamail for all her administrative support. PSI's mission statement is improved health for all Ontarians, and they provide funding to support speaker series such as this, as well as trainee sessions. So again, a huge thanks to PSI. Now, before I introduce Dr. Malkin, we'll start with our land acknowledgement. Ottawa is built on unceded Algonquin Anishinaabe territory. The peoples of the Algonquin Anishinaabe Nation have lived on this territory for millennia, and we honor them in this land. Their culture and presence have nurtured and continue to nurture this land. CHEO also honors all First Nations, Inuit, and Métis peoples, and their valuable past, present, and future contributions to this land. So it gives me really great pleasure to introduce Dr. David Malkin. Dr. Malkin's a professor of pediatrics and medical biophysics at University of Toronto. He holds the CIBC Children's Foundation Chair in Child Health Research, is a senior staff oncologist in the Division of Hematology Oncology, director of the Cancer Genetics Program, and a senior scientist in the Genetics and Genome Biology Program at the Hospital for Sick Children in Toronto. Dr. Malkin co-leads the SickKids Precision Child Health Initiative. He is co-director of the SickKids Cancer Sequencing, also known as the KICS program, which integrates and translates next-generation sequencing into clinical care of children with cancer. And he's director of the Pan-Canadian Multi-Institution Precision Oncology for Young People Profile Initiative, of which many of us at CHEO are involved with. Profile is establishing a pipeline to incorporate next-generation sequencing into novel clinical trials to provide precision oncology for children and young adults with hard-to-treat cancers across the country. Dr. Malkin's research program interests focus on genetic and genomic mechanisms of childhood cancer susceptibility, which he has explored particularly in the context of the TP53 tumor suppressor gene and Leaf-Romani syndrome. Recently, his work has addressed the application of genomics to develop rational clinical surveillance and treatment approaches for children and adults at genetic high risk for cancer. And on a personal note, David has been an inspirational mentor for me for many, many years, since I was a medical student in Toronto, and he's a close collaborator and a dear friend. The title of his presentation, Precision Child Health, Transforming Pediatric Care from the Genetic Code to the Postal Code. David.
Thank you very, very much, Jason, for that wonderful introduction. I really appreciate it. And it's wonderful to be here back in Ottawa. Ottawa is actually my hometown, and I always love coming back, even if the weather isn't so great today, but we'll forgive you, Jason, for that one. And it's also a little weird, of course, as everybody knows, sitting here at CHEO, actually just around from Jason's office at the moment, giving this Grand Rounds virtually in person. I suppose, with the background of our one of the floors of the SickKids Research Institute. So I'm not quite sure what that juxtaposition means, but I think it goes very much to the importance of the collaboration that I'll touch on and that we know between our two institutions, and looking forward to many more years of really exciting advances with us. So I have taken this title actually from a comment that Ronnie Cohen, who is the chief of the CEO at SickKids, a geneticist and a wonderful friend, the concept of the genetic code to the postal code, reflecting precision child health or precision medicine. And we'll see as we go through this why that phrase is so apt. So I think you can see my screen, I hope. To try this again, it's not seeming to advance.
Yeah, we can see your screen.
The question is why it's not advancing. I'm just going to do something here. Okay, so this is really the outline of what we'll talk about today. I'm going to give first an overview of our consideration of precision child health's definition and vision, a pathway of where we currently are and where we'd like to be, and then 6 vignettes, if you will, or spotlights of different concepts in precision child health, and then going forward in terms of how we address the challenges that we see. And on the right-hand side of the screen, we see really this amazing core team. Dr. Sobrero, PJ, is a respirologist, Associate Chief of Research at SickKids. Many of you will know her as she co-leads this with me, and there's a complementary area of scientific expertise and clinical expertise. Ariska and Thomas are program managers and doing brilliant jobs in many, many aspects of the program to continue to move it forward. And Yael is a program coordinator who similarly has made sure that we keep our eye on the ball. So the 2025 strategic plan for SickKids was unprecedented outcomes driven or empowered by precision child health. And so from the perspective of our hospital, at least, PCH is front and center to everything we do, with the concept being that within the next 10 years, probably realistically, it simply becomes a culture of the institution. We're nowhere close to that yet, as we'll see, but that's really ultimately where we want to go, so that all the projects, all the concepts that we develop over the coming years will just gradually become the fabric of the practice of pediatric medicine going forward. And this is a very complex figure that is used for board meetings and so on, really just articulating the many 6 sort of major aspects of the strategic plan, and just highlighting in yellow here the 2 individualized child and youth health care, and then building an academic health sciences center for the future as being those that are most relevant, if you will, to the PCH agenda. The current status, of course, at SickKids and pretty much everywhere else is this. I mean, the data that we collect across the board is fragmented across various databases, and even though we have Epic in place, I think as you all probably would agree, at the moment it's really not that we're not that confident that we can extract and collate the kind of data that we need with that alone. In addition to that, most genomic, environmental, and other key data is not routinely collected on all patients. It's collected in different cohorts of patients depending on specific studies, but not as a general rule. Targeted therapeutic options are rarely available and even less so accessible to pediatric patients for many reasons, not or perhaps most strikingly being the challenge of getting drugs into kids, period. And then, of course, opportunities to connect patients to research are really siloed. They're based on specific studies or diseases, and so there's no institutional opportunity, at least at the moment, which we'll touch on a little bit later, as to bringing everything under one kind of umbrella. And so because of that, really, there are silos within the organization, as much as we'd like to really believe and want to know that that's not the case. So these are some of the challenges to try to face. Now, Dr. Peter Jatmari, who's the past chief of psychiatry at SickKids, chatted with me for a while before this grand rounds and presented some concepts of what really is precision child health. And we know that it comes from the discovery of cancer genomics, and that's been a feature, if you will, of the traditional approach to precision medicine. But we know as well that while targeting molecular signatures has led to some remarkable outcomes, cancer being one of those particular areas, the thought is, are these better than the treatments targeted to the disease itself? And we believe that's the case, and we believe that's why we should be pursuing this strategy. Really, it has become the paradigm. It's sort of the buzzword. Not that long ago, we talked about personalized medicine, and that has shifted to precision, because I think as physicians, we tend to want to believe, and I think we are constantly thinking that we do practice personalized medicine with each of our patients. And it, as I mentioned, is now beyond the cancer genomics. And this is where the concept of from the genetic code to the postal code comes in, where we start taking into account many, many aspects of the child or the family's health. We have a significant reliance on static biomarkers, such as the various omics signifying targets, because that's where the science is. And we need to increase our understanding of pathophysiology of disease to better appreciate how we can use precision child health. So the principles that we think about then are that PCH would integrate all information about a child's determinants of health, from the genome through the environment, and also the preferences, meaning really what are the ones that are unique to a particular child, or how do we come up with an algorithm that is unique to a particular child as well. In addition to that, we are taking advantage of bioinformatic and computing tools much more effectively, and both our institutions and many others have great strengths in AI and machine learning to develop more effective opportunities in preventive actions, diagnostics, and therapeutics. And then, of course, at the end of the day, this really ultimately, we hope, would fuel the next generation of preventive care. I mean, really the holy grail, if you will, is to prevent childhood illness rather than to always kind of being reactive and jumping in with diagnostics and therapeutics. But there is this balance, because up till very recently and still now, we really talk about evidence-based medicine. And I know really in my training and especially through the 90s and 2000s, the concepts of evidence-based medicine really were very powerful and very important. The question is, does precision health and evidence-based medicine conflict or conflate? And this argument, I think, can be developed in many different ways. And it's, I think, important that we understand that, because evidence-based medicine is really based on the premise that one collects large amounts of data in a particular problem, and then uses that to derive a solution for an individual patient. Whereas precision medicine, really, you're talking about the specific patient. Yes, you're collecting large amounts of data at the population level, but then you're trying to say at the same time that the individual patient does not reflect the mean or the average or the median of that large amount of evidence. And I think all of us in our various disciplines in pediatrics recognizes these challenges, where one looks at a large scale clinical trial of, let's say, treatment of X or Y with drug Y or Z, and then you decide to put your patient on that trial, not really knowing whether that patient will or will not respond as an individual, because the trial really is just reflecting the population. So with some of those caveats in place, then, we've developed this 10-year plan, as I mentioned, for the Precision Child Health Initiative at SickKids. It was launched in 2020, literally a month before the pandemic struck, and there was a big excitement about this. Even the naming of the new primary hospital, which still has yet to be built, was principled around the concept of a structure that also facilitated precision medicine. And then as you can see here on this roadmap that we're heading towards launching a funding campaign. Some of the homework is being done, a lot of it now already for 2023, which coincides approximately with the end of the campaign for the new building. And then of course, the subsequent number of years will be ultimately to implement it, as I mentioned, as a culture of our institution. The current state of precision child health, or I should say the current state of translating discovery to translation into clinical care, is really a long and winding road, to quote a Beatles song. And there are many different aspects. There's multiple forms that need fill out. You need to design things. You need roadblocks along the way in terms of budgets and so on. There's a lot of scientific and operational review. There's ethics review. And eventually you get there. But as I think we all know, there are times, especially in Canada, where investigator-driven trials from our institutions have been initiated. But by the time we get to the complex road of getting them into patients, the trial is closed and all the patients were enrolled at other institutions, very often in the States. So we need to really speed up the process, and the process will be sped up by a much more integrated approach to knowledge management, data management, and change management, which we'll talk about. Another way of looking at this is really to, again, keep the patient-centered approach. And so we think of having our patients and families as active participants in their care, of course, but also that does then require a much more seamless and open approach to consenting patients, to be able to generate data from them and keep that data in such a way that it will be available for the development of these large-scale projects that will benefit all patients at the institutions and at our institutions across the country. So this sort of circular generation of data and then using that data for individual patients is ultimately what the journey will be. So we've created this sort of timeline, if you will, or sort of kind of a timeline. It's a lifeline where one would be generating data at a prenatal all the way through to late adolescence, but very importantly that this data should be captured in such a way that it can also be developed and translated for when patients transition out of our institutions into adults, because again, this is one of those challenges that we face, is that we know kind of what happens until they leave our institutions, but after that, the system tends to be very, very fractured, and we don't really know how well we are doing when it comes to developing novel approaches to treatment of children that will benefit their long-term outcomes as adults. And so this is an area that we think we need to continue to keep focused on. And to do that, at least the way SickKids, we've sort of thought of this as a bit of a puzzle where we have discovery research based in the Research Institute, which is often very fundamental. It's translated to humans as a general population, and then back into specific patients at an individual level, translate that into clinical practice within the institution or partner institutions if these are collaborative efforts that ultimately lead to a standard of care at the hospital level. And so the bridge is this ability to integrate basic fundamental discovery through clinical research into hospital practice. And this is a very complicated slide, but just sort of shows some of the different aspects of activity that are required to make this happen, all the way from developing really well-integrated clinical operational units. Ethics and REB will talk about this, because the idea being that if we are going to start thinking about universal, quotes, consent for patients to be enrolling on pretty much any research study that could benefit them or others and lead to standard of care, then we have to rethink the way that legal and regulatory and ethics approaches are taken in the concept of precision medicine, precision child health. And then at the end of the day, what this really means is developing these large-scale platforms, developing zebrafish avatar models for Jason that could immediately be translated into clinical care. Translational genomics, both institutions doing extremely well, but we can do better there, and so on. So this is a complex thing, and that's why we really envision it over many, many years. And as an example, if you did take one individual, we're talking about many levels of data across the institutions from other institutions being captured in such a way that they will immediately inform an individual patient's care. So for the next few minutes, I'm going to touch on 6 stories, if we will, or spotlights or vignettes that capture different aspects of the precision child health concept. So the first is actually co-led by PJ, and this is the Child Initiative, which I think many of you will be familiar with, a normative cohort of several thousands of patients across the country. And this really has been an incredible initiative because what is being captured are multiple levels and multiple types of exposure or genetics. A number of these patients, and several hundred, have had whole genome sequencing done, and then starting to use that data to try to be more predictive of outcomes across the population. The other thing, which is very important for any of these sorts of studies, is try to set them up in such a way that the future what might not be predicted can be captured. And so one example here is we put in the COVID-19 pandemic. I mean, this was not envisioned really by anybody, I'm sure, when Child was developed, but it is designed in such a way that data on COVID exposures can be captured. And of course, from PJ's perspective as a respirologist, and one large part of the cohort are patients with asthma, this would be an incredibly important aspect. So as we think of how we develop precision child health projects, we have to think of them in such a way that all data and new data elements that are not previously foreseen can also be integrated. Now, what this also takes advantage of by being the kind of cohort it is these 3 main areas. There's genes, which are presumably immutable in the sense that you're born with what you're born with, although there are going to be epigenetic changes and epigenomic changes, which are influenced by both the environment, which you may or may not be able to avoid, as well as individual patient choices, which one may be able to avoid. And so having such a way that you can integrate all this data at a patient-specific level is incredibly important. And it's also now, of course, emerging, not just in the concept of asthma and respiratory disease, but many human diseases, that the microbiome is playing a much sort of stronger, if you will, influence than previously, perhaps 5, 6, 7 years ago. We hadn't really thought through that well, at least conceptually. And one of the things we were chatting at dinner very briefly about last night is, if you look at the extremely old literature in the late 1800s, very early 1900s in the cancer field, patients with cancer who develop overwhelming infections of one sort or another, and pre-antibiotic era, it turns out that if they did survive the infection miraculously, it was not that infrequent that their tumors would have been seen to regress, maybe not be cured, but definitely regress, suggesting that actually the microbiome, microbes did have some sort of influence even in that area. Now that, of course, is very rudimentary, and we now have a great deal of information of how the microbiome may be influencing a whole spectrum of disease across the lifespan. And these are kind of the ways that we start thinking about it in promoting health. So taking advantage then of such a large scale cohort in child, and then thinking of how can we emulate that in disease cohorts. And there are definitely disease cohorts across the country and around the world that are similar to this sort of normative data set. But the problem is, again, that they tend to be scattered. The data is not collected in a uniform fashion across countries or even across cities. And also the concepts of data sharing, which are very important because most of the pediatric diseases are relatively rare, needs to be addressed so that having data is in and of itself not sufficient unless you can actually access it, share it, and then explore it. So a second approach is the concept of AI. And so for this one, I am definitely not an expert in AI and machine learning. We do quite a bit in our own lab in the concept of cancer predisposition, but I rely on my colleagues and frankly, 3 of my students who are brilliant mathematicians to guide me through some of the nuances. But Devin Singh, who is in the emergency room at our place, and again, I think many of you will know him, has been using AI to address some significant problems in this case, in the emergency room. And I'm sure like your ER, our ER is absolutely packed with patients and families basically spilling out, waiting all over the main foyer, if you will, of the hospital. So to address this and try to reduce adverse outcomes associated with that, Devin looked at it this way and sort of studying what the current workflow is and noticing along the way that from the time a patient enters the emergency room and is triaged, there are multiple steps along the way before they actually exit. And this can take, even this initial point to having a first assessment can take several hours. And then, of course, depending on what their ultimate sort of evaluation is, this time frame could be another several hours. And so clearly lengthy wait times are not good for the patients, they're not good for the system, they're not good for any of the healthcare workers. And there have been many attempts over the years to try to figure out how we reduce that. And so what Devin has been doing is developing a project where they're collecting all of the health record triage data, identifying the demographics, vital signs, symptoms, the triage notes, processing this in such a way that you have large enough amounts of data across a large enough cohort of patient, then using machine learning models to try to predict upfront which types of patients based on this initial data entry are more or less likely to require immediate or longer term help within the ER. And by doing this, it's actually quite fascinating because they've been able to already reduce test ordering for about a quarter of the patients in a relatively short period of time. So that initial step here from entry to here is being reduced significantly, which is critically important. Now, one area that they're also very interested in exploring in their studies going on is to actually have a lot of this data collected at the time that a patient may call to the ER or call their family doctor to be referred to the ER, so that as soon as they hit the triage note, all of that information is already ready for them, and then it sort of advances the queue much more effectively. So the use of AI in improving access is incredibly important. It will lead, we believe, to faster time to diagnosis, which of course applies to many areas of medicine, and will take advantage of many of these other aspects that we've touched on that are not necessarily available at the time that the patient arrives in the emergency room, but may be very relevant to how long they actually stay there. Now, going along the same route, this is work that is led by Jai Mazawi, who is one of our ICU docs, and also a great deal of expertise in the AI world, came from prior training in Cleveland, in Cincinnati, and also in Boston before he arrived here a few years ago. And the concept there, of course, is that we're kind of sometimes scared, I guess, about the implications of artificial intelligence and this collection of large-scale data. But in fact, we all know with every device and instrument that we use that there's plenty of data already sitting out there, which really is reflecting on our own on our own lives. But the question is, can we use that for improving things? So bringing up the Tesla as an example, and the Elon Musk story is, can you create a device, in this case a car, which pretty much does everything based entirely on AI, that you can take the human out of it? And I just saw a story a couple of weeks ago where apparently somewhere in California, a police officer pulled over a car, I guess a auto-driven car, because one of the headlights was out, and notice there was no driver. So I'm not quite sure how you give a ticket to a car that doesn't have a driver, but it was kind of interesting point. So here, of course, we have the capacity to build things like this, which are completely autonomous. And they're so autonomous that, we've seen this picture. It is now pretty much standard in most cars to have multiple sensors around the car to be able to drive not only autonomously, but of course, when you're driving it yourself, there's different beeps for every possible direction that the car is moving, what's coming at you, what's not coming at you, and so on. So if this can be done in this context, the question is, can we do this in the healthcare context? And of course, the answer is yes. One of the challenges that we always face, I go back to the days where you say, well, if you can put a person on the moon, why can't you ... cure cancer, cure heart disease, cure pediatric asthma, and so on. And we know that the technical engineering that goes into these sorts of discoveries is spectacular, but there is no real biology here. And biology is much more complex, and of course is the aspect of applying AI that makes it interesting, but also difficult. So this would be perhaps a typical, and maybe a slightly over typical example of a baby in the ICU with a multitude of instruments measuring everything on that baby, and you basically can't see the patient for the instrumentation. So how do we start thinking about this? Well, we know in the medical imaging focus, and many of our medical imagers really taking some exciting new approaches using AI to better articulate and distinguish not only where lesions are in this case in the brain and an MRI of the brain, but also to be able to use a variety of tools that are AI driven to improve diagnostic accuracy and then guide our surgeons or guide our medical care into determining how best to approach these things. And so this is now becoming really standard in many contexts, and of course the technology continues to improve. In the context of the ICU, Zai has demonstrated beautifully this interesting concept that could you predict, based on heart rhythms in real time, the likelihood of a child having a cardiac event, a cardiac arrest, prior to the event actually happening. And this, of course, is critically important, because if that's the case, and if you can time it in such a way that there's at least a few minutes, the entire team can be ready, even if it might not be possible to prevent the event, to actually intervene and reverse the event when it happens. And so this is basically just complex algorithms, which I won't go all the way through, but still has that same principle that it requires large data sets, which are processed, and then ultimately using machine learning tools are able to classify a rhythm. And by so doing, you can actually identify whether the individual patient has a normal rhythm or is entering into some atrial fibrillation or other rhythm abnormality. And in that regard, that pings on the one of the tools, and one is able to intervene. The next example is looking at pharmacogenomics, and this has been an area of work for many, many years, but is now really starting to find its home in a multitude of areas. And Iris Cohen leads this effort at SickKids, and some of the examples here were taken from specific examples. A young boy who had been on the Berlin Heart developed blood clots despite being on clot prevention meds. And so pharmacogenetic testing was done, and it turns out that this young patient had been a poor metabolizer of the CYP2C19, and was not able to convert this particular drug to its active form. And so because of that, child was switched to aspirin, and the clot was reversed. And now it's basically standard practice to do this for all patients who put on the Berlin Heart, and several have actually been reversed, had changed. And this, of course, these results can come back in very fast real time, and so you have an immediate effective care. And this is a true example, I believe, of PCH. Another example is in the context of eosinophilic esophagitis, where here's an example of a toddler with food allergies, eczema, and so on, diagnosed with this particular illness. And then here again, pharmacogenomic testing was able to identify a variant which would predict that standard medication and dosing would not help, and so you can actually switch the dosing and switch medication as required. And this is really the way that we like to think that we should be able to do medicine. One of the key principles, of course, is rather than starting them on the, quotes, wrong medicine first, would be to be predictive and start them on the right medicine right from the beginning. So you're not switching, you're actually starting from the beginning. And so we know this to be a tool that is now being applied across our institution and yours and others, this whole area just continues to expand as we understand more and more about the pathophysiology of disease. This particular example, Jim Dowling, who's one of the neurologists and neurogeneticists at SickKids, has a great interest in the rare genetic diseases and rare neurologic genetic diseases. And this is a example of a different way of approaching how to solve the problem. So one of his young patients has SPG50 and a very rare disorder, the only one in Canada. And is a progressive disease with moderate intellectual disability and progressive and ultimately fatal. So this young lad's parents created a organization or a site and have been working diligently and incredibly passionately with FDA, with Health Canada in North America, to see if there was a way to develop a gene therapy approach to correct this gene defect, recognizing that he is already, I think 3-4 years old, and so the benefit to Michael himself might be limited, but the potential benefit to future children, and probably even most importantly to many other children with similar single gene devastating diseases, could be enormous. And so this particular setup has been driven very much by the family, but it has engaged industry as well as multiple institutions. And recognizing this fact that, I started bringing coals to Newcastle here with Kim Boycott and her team in terms of rare genetic disease, that there is a high frequency of rare genetic disorders, and finding the specific therapies is, of course, a big challenge. And many of these are, of course, enormously expensive. So addressing that problem will be important. Now, the pathway to cure, if you will, is very interesting because the base biology was sort of understood in about 2 years ago. And then with the development of the particular vector, and this was a relatively fast in a couple of years to complete that. And then another plan, of course, to be able to have the drug available to infuse. And in fact, the child did have this infused just, I think it was 3 weeks ago, had his first infusion, which is really pretty remarkable. And there's, I gather, going to be quite the story about this. And now the plan is to start to expand this out. So the process is in place. Now can we expand it even further? Now this 2-year timeline might seem relatively long, but in relative to what it used to be, if one thinks of, for example, Gleevec or imatinib, one of the classic tyrosine kinase inhibitors for certain types of cancer, that was about a 20-year time frame from discovery, probably even more, from about 30 years from discovery of the fundamental defect to an actual drug, and then ultimately the clinical trial. So we are getting better at accelerating discovery to clinical approval, but it's still, for many patients, of course, not fast enough. And then the second to last vignette will be in the context of the partnership in terms of the SickKids Precision Child Mental Health Initiative. Now, I am not a psychiatrist. I'm not expert in mental health at all, but this is really a remarkable advance and really reflects in many ways what we need to do, both as two institutions and ultimately, I believe, say at a national level, if we're going to really make precision medicine and precision child health the quilt, if you will, that we want it to be for the way we practice medicine. So I'm taking an example that is shared with me both by Peter as well as Steve Shearer of autism spectrum disorder, which from a historical perspective is really, I find, quite fascinating. My area is cancer genetics, and so there's a lot of overlap here in the way we do things. But, you know, what's interesting, as we'll see with the last scenario, is in the context of autism spectrum disorder, there's a heterogeneous phenotype and clearly a heterogeneous etiology. But for the most part, with very, very few exceptions, there aren't as yet targeted genes that explain many of the, or very few rather, of the cases of ASD. Whereas at least in cancer, we actually have a lot of targets. The problem is trying to match the target with the drug or match a target with the patient. So let's take a look at this. In the context of autism spectrum disorder, we see that there are some molecular pathways. We'll see very shortly that in cancer, it's quite a bit different than this, and selected variants that are associated with this. And this is work that has been done by many people, including Abdokia at our institution. And what we're thinking of, again, when we take genetic or genomic information, is to identify whatever disease entity it is. There's a certain number of variants. They have a certain phenotype associated with them. And then one starts to think of specific treatment protocols, which might be targeted agents, or they might be agents that modify the effect of the genetic event. And then accumulate this over large data sets to start to better interrogate pathways and figure out which pathways are relevant to which specific patient. And so this is work that is set to be submitted by Steve with a large number of collaborators, of course. And really, this slide is not meant to be read other than to show that there are multiple data sets being used and then interrogated to ultimately lead at the bottom here to specific genotype-phenotype correlations. Now, one of the things that is outstanding in this context is, as you see here, this is taken from 3 different data sets, and there are over 20,000 patients encompassed in these. And this is how you're able to actually generate amazing data. This is, in our world in cancer, it's very hard to generate those numbers in the pediatric space. So we sort of envy the ASD work. And with this, using the 2 large data sets here, we see, as I mentioned before, that very few of patients with ASD actually do we have a potential rare variant detected. Most of them really are still undetected. And then there are multiple, when you take the subset of this 15, there are multiple different pathways and targets which are identified. And they sort of, depending on the cohort, fairly similar spectrum. Nonetheless, even though they're relatively rare for some of these, the genome sequencing can lead to specific patient-specific indications for clinical care management. And these are sort of 2 examples that are just highlighted. Okay. So the last highlight then, or last spotlight then, will be cancer. And this is really, if you will, the problem that we face. They are rare diseases, even though cancer in and of itself is the most common cause of disease-related death beyond the newborn period in children. Each type of cancer is relatively rare in terms of numbers. The genomics is very complex and very different than adult onset cancers, for which there's a lot of data. And drug access, like with all pediatric conditions, is very, very limited. So unlike the autism spectrum disorder, where there are certain pathways that are being explored, in cancer, it's pretty much every pathway under the sun in terms of the way that developmental biology works. Cancer is essentially an alteration or an abnormality of human development. And so one constantly sees the accumulation in more and more pathways in normal homeostasis, which lead to the development of cancer. So this is just an example. You saw the pathway before for ASD, and this is just a picture just to show in cancer, it's probably several orders of magnitude more complex, which of course makes it that much more difficult to figure out what you're doing. So what is being done? Well, Jason had mentioned earlier, we have this sequencing program based at SickKids. We've sequenced now about 700 children with hard to cure cancers. This is Marlow, who is actually one of my patients, and sadly, she passed away a few years ago. But what her course was that she had several different chemotherapy drugs. I'm sorry, she had alveolar rhabdomyosarcoma, which was metastatic at diagnosis. And she was told at the time that she was diagnosed that if she failed her initial therapeutic options, she really had no chance for cure or really no chance for further remission. Her parents brought her to SickKids and she lived another 8 years, but that was in part because of the intensive therapy. She had lots of sequencing done. She had several targeted drugs used. She had immunotherapy, hundreds of thousands of dollars spent. But what was interesting and very unfortunate is she actually had a druggable mutation in the TP53 gene. And the only problem, but it's a key problem, is that drug is not available in clinical trials for children. It's not even available from the company for compassionate release in children. And so she never really had an opportunity to see whether that would or would not have had any effect. She became essentially the poster girl for our hospital, for the new building, and for precision medicine at all. On the other hand, we've had patients like this, a young boy, Izaan, who had a large sarcoma not otherwise specified in the pelvis, seen here on the this MRI. She received standard therapy, which was vincristine, cyclophosphamide, actinomycin, and the tumor kept on growing. He was just about 10 months old at the time. He was unable to walk, constipated, significant bladder retention, as you can see. Also sequenced and was found to have a B-core tandem duplication, which redefined the diagnosis to a primitive myxoid mesenchymal tumor of infancy. Switch therapy, had an immediate response, and 2.5 years later remains in a complete remission with absolutely no side effects whatsoever. So it is amazing how you can completely change the course of a child's illness through these powerful technologies. And there's actually a story, parents talk about this here. But then now just finally bring it around to really the soup to nuts all the way through. And this is work that's done by my colleagues, Uri Tabori, Adam, and Eric leading this with Daniel Morgenstern on the clinical trials effort in the context of constitutional mismatch repair disease. Very rare disease associated often with consanguinity and biallelic mismatch repair defect, which makes these patients highly susceptible in early childhood to colon cancer, high-grade gliomas, and leukemias. But what Adam had shown a few years ago was actually these patients have a very high tumor mutation burden, meaning that they have tens or hundreds of mutations per megabase in the genome, which is way above what you would see in other pediatric cancers, or for that, most adult cancers as well. And that makes them potentially eligible for immunotherapy. But before we get to that, we think about prediction, we think about prevention. And so based on a protocol that we've done for Mifromeni syndrome, Uri and his colleagues developed one surveillance for early tumor detection using colonoscopies, frequent brain imaging, and show in this slide taken here that the overall survival for patients who were undergoing surveillance was significantly greater, this sort of top blue line in the Kaplan-Meier, compared to those who were not undergoing surveillance. So surveillance does an early detection at a tumor-specific level is important. But the other part, of course, is you identify the specific variant or alteration genetically, and then you identify a therapeutic intervention, which in this case was immune checkpoint inhibitors. And what we see here is that in patients who are receiving immune checkpoint therapy, their overall survival is actually significantly improved. In this case, these are the patients with brain tumors, glioblastoma, than it would be historically. And GBMs are notoriously virtually incurable or extremely rare. And this team at an international level is now seeing patients who are having long-standing remissions. I don't know that we'd all call them cures, but they're remissions well beyond 3, 4, 5 years. When we look at it at our whole program, we're finding that some 45% of patients who are sequenced using the KICS program are found to have a therapeutically actionable target, which is very exciting. And in fact, about 2/3 of clinicians are finding that one aspect of the genome profiling, whether it's germline or somatic, is impacting clinical management. But the challenge is you can find the mutation, and this is a very complex diagram, and it's meant to be that we find a molecular alteration, but then we don't always have drugs available. And so we're now having to develop these compassionate access, working with Health Canada, future basket trials, international collaborative trials, and so on. And even with that, we only find a handful of drugs that could be made available to these kids. And so that's one of the critical aspects of precision child health is, again, we can generate all the data, but unless we can get the drugs, and unless we can get the drugs into kids, I'm not so sure that we've actually completely succeeded, and that will be the future. So the final couple of slides, and some time for questions, is what is the investment? So of those 6 projects that I've identified, almost $90 to $100 million has been invested already. These are precision child health projects that exist before precision child health actually exists. SickKids Foundation is committing a significant early, sort of over the next couple of years to address some of our infrastructure needs, and the AI AIM program, artificial intelligence, a similar amount. But at the end of the day, we're probably talking in the neighborhood of 50 to 70 billion at least to really have a program like this going. But as I said, ultimately, if this becomes the fabric of how we practice medicine, then this should, one would hope, be covered entirely by global budgets for institutions, and that's a whole other discussion, of course. Until that, we have to solve these problems. We need to identify why there is a lack of interoperability, and we need to better understand how to address that. As I mentioned, we need to develop new ethical and legal ethics and legal guide frameworks and regulatory policies to change the way we practice. And of course, the current funding models are not sustainable because they're currently philanthropy and research dollars, and so a shift has to be had there. Ultimately, though, these are the key questions, and I just focus on this one, which is taken from Peter Jagmari, is whether the incremental potential benefit and outcomes by incorporating these precision health strategies is actually worth the extra cost. And this is, of course, what we need to fundamentally understand. At the end of the day, will the investment in PCH ultimately lead to what we want, which of course is the improvement of lives. We believe that to be the case, but we actually have no evidence at the moment, and so that's where a lot of the new research will go. So with that, we'll look to the future, I just put this up because we think that precision child health is the holy grail, but we're sometimes not quite sure what we're looking for. And I will leave that question at hand. And thank you again, and look forward to a few questions. Thanks.
Thanks very much, David. That was a wonderful tour de force, and really showing cross-cutting themes of precision child health across numerous areas. So we do have a couple of moments for questions. So I see Dr. Abbott has her hand up. Leslie.
Thanks, Dr. Berman. Hi, David, Dr. Malkin. Thank you for coming. Loved your talk. I wanted to just touch back on your title a bit, where you talked about postal code. And I know with my involvement with you in the profile project that this is a passion point for you too, of making sure there's access across the country to precision health. And I'm just wondering if you could speak a bit to that, and maybe in particular to the profile project, how we're making sure that kids across the country, whether they're at SickKids or CHIO or wherever they are, have access to the same precision health.
Great question. Thanks, Leslie. So I think that we have to be a little careful of the translation. So within the context of cancer as the elemental disease, let's say in profile, because the vast, I would say, almost 100% of patients, regardless of where they live, if they have a diagnosis of cancer, will find themselves in one of the 16 pediatric oncology centers or an adult center. then their access is less of an issue than I think it is for many, many other diseases where there's not such an integrated network of institutions that have developed over years for cancer. So I think there can be lessons learned from that, though. I think what it means is that we have to develop, as part of this whole precision access, is much better relationships between the family practice units, the other subspecialty units, our adult centers for these older adolescents who might find themselves in an adult center, let's say like Ottawa General versus CHIO, or in our case, Toronto General versus SickKids. That's where the solutions will come, and it is a big challenge. I'm not sure we know what the specific solution is. So cancer might be an exemplar for other disease entities, but very, very important point. Thanks.
Thank you.
Thanks. Dr. Hadjigenassis.
Hi, thank you very much for a great talk. I just had a quick question, and I might be thinking of this oversimplistically, but when I think about precision medicine, it feels like we're taking multiple variables from a particular patient and putting it through an algorithm to sort of give us a more I guess a more precise care plan. I was just wondering about your thoughts on potential biases created by these algorithms or algorithmic biases, and what we can do to mitigate those.
Yeah, thank you. I think that is a big challenge, and it's because sort of alluded to, in the pediatric space, even the most common disease is relatively uncommon compared to our adult colleagues. So the numbers become an issue. And people like Anna Goldenberg, who leads the AI program here, or Jai, or Devin, or all these people, will say the same thing, is that if you don't give us enough data, we can't develop the algorithms that are terribly useful, because the biases just constantly keep on coming up. So that is where examples of partnership, like we've got here between SickKids and CHIO, expand that to ultimately a partnership right across the country. So we have a natural partnership, and you just accumulate more data. The more patients you have, the easier the algorithms are able to be developed, and then you can apply those to individual patients because you start breaking down individual data elements. But you're absolutely right. I think that the child cohort has been a good example. I think the autism cohort has been an excellent example, but that one is being done because it's international, right? I mean, they've got thousands and thousands and thousands of patients. So they're going to be able to generate the kind of data that I think many of us in other disciplines are going to envy. So I don't know if that's really the answer, but you've identified a key problem going forward. And I know our AI people struggle with that a great deal.
Thank you very much.
Last question to Dr. Maharaj. Wendell?
Thank you very much, Dr. Malkin. Excellent talk. This strikes really very much at the core of something I've been struggling with my entire career, which is basically, and as a cardiac surgeon, we do a number of standard processes on essentially every time a non-standard unit, and are surprised when we get non-standard outcomes. So I very much see the application of these things for rare types of diseases and subtle nuances in genetic coding. Do you think there's a scope for this in terms of a more utilitarian approach to being able to predict specific outcomes after things like surgical interventions, like cardiac surgery, that we do in a fairly standard this model of provision?
Yes, I believe there is, and I'll say I'm not a surgeon, so that this is spoken with the wisdom of the non-combatant. But what I'm hearing from our surgical colleagues, people like Jim Drake, for example, who is the head of surgery, one of the neurosurgeons, and also works in the area in terms of trying to design new ways to approach the problem, is that there are ways to apply this. And one example would be, as you're probably familiar with, the black box in the OR, the concept of collecting data in every operating room, in every operation. So if you're, as a cardiac surgeon, if you're doing the same procedure in multiple patients, say Fontaine, whatever it is, that you collect everything from the temperature of the room, from any interruptions during the surgery, from conversations, from the types of stitches, from so on and so on. Again, you do that with multiple things. The hope is that you'd be able to then predict ahead of time where the faults are, if you will, with the process during that whole procedure that may be predictive of outcomes. So that's from the technical perspective, there's some great value. In terms of the outcome, in terms of the patient, assuming everything goes well during the surgical procedure, same principles apply. I mean, the question would be, what is the underlying fundamental pathophysiology of the disease in that individual patient? What was the surgical intervention? How well did it go? And then using all that data to predict what the downstream piece will be. So our surgeons, and I'm sure you as well, are extremely excited and interested to see how they can apply these principles for sure.
Thank you.
Thanks for the questions. David made reference to the Shared Precision Child Mental Health Initiative between CHIO and SickKids. I think many of you have heard about this. We're very excited about this process and an initiative. The team is being led by Kathy Pager, Christina Honeywell, and Heather Howley. And this is a plug for the launch event next week, May 5th, at a special psychiatry grand round. So hopefully you'll be able to attend. I want to thank everyone for their attendance today. We had a record number. At one point, we had, I think, close to 180 people on the call. So thank you. I think that speaks to people's interest and engagement in this topic. This was recorded. So for those of you who weren't able, for those of you who had colleagues that weren't able to attend, please let them know they will be able to access this content on ChioNet. Dr. Malkin will be here for the day as part of the PSI series. He's leading a workshop and a trainee session and having some one-on-ones in person later this afternoon. So I want to really thank David again for coming. This is the first time we've had someone come in person in a very long time. So really nice to be able to host someone in person and to thank him for his really inspiring talk. I want to thank all of you for your attendance. Again, I want to thank PSI for sponsorship and our PSI committee for selecting David as a speaker. And have a great day, everyone.